genome regulation. Started his PhD work as a graduate student in Eric Lander's group, after which he then directly took a faculty position at the computer science department at MIT. And in the last 20 years, he has made numerous contributions in the annotation of regulatory elements in the genome as part of both the ENCODE consortium as also as part of the roadmap epigenome. In fact, he co-led the epigenome effort and published a highly influential paper on the 111 epigenomes. That was almost 10 years ago. And he has also been a driver in evolutionary comparison of genomes. And now the most recent studies of single-cell genomics has been really a leader in all fronts. So we're going to hear from that. I want to tell you a little bit about something that you probably don't know. So besides Manolis being such a wonderful scientist, he is also, I understand, a what you call the architect. His family has really a tradition of building houses. And when I visited his house a few years ago before pandemic, I was amazed to learn that he himself designed a new house, broken down and restarted, building up from ground almost. And it was a beautiful house. And I know something that I don't have, among many things that I know, that I admire Manolis for many other things. So without further ado, welcome Manolis.
Thank you very much.
Floor is yours.
Thank you. I really want to thank Bing for hosting me today. I'm here for a meeting on Alzheimer's disease tomorrow, and I'm like, I can't come to San Diego without seeing my favorite people. And I first visited San Diego when I interviewed for a faculty position here, and I was very kindly offered one about 20 years ago. And I told them, mark my words, I will move here one day. And every time I come, I'm like, yeah, I still think I will move here one day. So anyway, I'm also very fortunate to just be in this unbelievable field, to sort of host friends from so many different labs in my lab. If you guys are looking for postdoc positions in Boston, we're always hiring talented people. So I'm always excited to sort of meet talented students and collaborate. So my first paper actually back in '99, I think, was on computational geometry. So 3D spatial organization of genomes, of houses, and of topological objects is one of my passions. So that explains, I think, the architecture. But what I'd like to tell you about today is our efforts in translating genetics and genomics into therapeutics. And specifically, how do we understand the circuitry and how do we manipulate the circuitry of human disease at single cell resolution? So this is really about circuitry. It's not about genes. And I will explain why shortly. So as you know, genetics is not a recent realization. Humans have understood the power of genetics for thousands of years. So selective breeding of animals has led to all of the domesticated animals that we have now, including, for example, going from an animal that will eat you to someone that can't wait for you to get home, from a plant that's completely unchewable to a major source of food here in America. And even for humans, the inheritance and the similarity between parents and their children in terms of eye and hair color, in terms of height, in terms of many physical attributes, and sometimes even personality is something that has long been understood. But it's not until the mid 1860s that Gregor Mendel showed that inheritance is in fact digital, that it is discrete, that there are discrete units that we now call genes, which are inherited in what we now call Mendelian fashion, where you either get it or not between parents and offspring. And what he showed is that for the phenotypes of peas that he was growing in his garden in Brno, which I actually was very fortunate to visit. I received the Gregor Mendel medal a couple of years ago. So I went back to the monastery and I brought some of these peas, which are now growing at my kids' school near my house. And all the kids are now doing experiments with those, which is kind of cute. But basically, what he showed is that for his P's, he could show that there are distinct phenotypes. For example, the color or the size, which are inherited at 100% or 0%. And also, the second rule was this concept of dominance and recessivity, that if you receive either one of your two copies from the dominant allele, you will have the phenotype. And if you both are recessive, then you will not have the phenotype. And his third rule was independent assortment, that basically every single locus, every single one of these phenotypes is inherited independently. And there's some debate as to whether he had to actually fudge his data to basically get these, exact numbers to work out, because some of the loci that he was studying were in fact slightly closer to each other than independence. But his work remained obsolete and obscure for about 50 years. No one built on it. It was only covered in a local newspaper when he presented it, and no one read his paper. He sent his paper to Darwin, who was at the time working on the theory of evolution, and was missing a key component for how inheritance actually works. And that would have made the theory of evolution so much more complete. But in fact, that paper was still remained unopened in Darwin's desk at his death, unfortunately. And why is that? Because at the time, people saw phenotypic variation in humans as continuous, not discrete. Eye and hair color that I mentioned earlier were in fact not just blue or brown. You had a whole continuum of hair color, of eye color, of height. And this was very hard to reconcile with the seemingly discrete inheritance of P's. And it's not until the beginning of the last century, in 1910 or so, that Ronald Fisher and many other statisticians basically worked, like rediscovered Mendel's paper, and then worked out that, in fact, with Mendelian inheritance of even 5 independent loci and additive effects, you could get a seemingly continuous distribution of height, just because there's a Poisson probability of inheriting more and more of these alleles that make you taller or shorter, ultimately leading to such a distribution. And the deviations that I mentioned from Mendel's law basically allowed us to start mapping Mendelian traits over the 1980s, showing that, in fact, the deviation from independence is directly proportional to the distance, or more like to the proximity of genes to each other on the chromosome. But it's not until the sequencing of the whole human genome that we could finally get at these complex traits that were governing human variation and polygenic diseases and these complex traits, okay, complex diseases and traits. So with the human genome, we were able to finally build maps of human variation, catalog all of the common variants in the human genome, understand all the haplotypes that these variants are sitting upon, and build the tools for massive genome-wide association studies. And why do we need massive genome-wide association studies? Because these phenotypes are not discrete like Mendel was seeing. They're in fact very, very subtle. If you look at human height, there are thousands of variants, each of which contributes less than 1 millimeter to your height, statistically robustly, but less than 1 millimeter. So in fact, you vary more than 1 millimeter between the time you wake up and then the time you go to bed, because we kind of lose height throughout the day and we regain it the next morning. So these variants are, number one, causal, but number two, very subtle in their effect. And number 3 and 4, I'm going to mention in the next slide. Why are we excited about genetics? Because genetics gives us the promise of causality, to understand that there are correlations between the variants that you have inherited from your parents. And for example, here, body mass index, a measure of obesity, tells you that perhaps it's not just your fault, that it's not because you eat a lot that you have these variants. It's not because you're heavy that you have these variants, the other way around. So basically, causality is very rare in human epidemiology. Throughout the centuries, human epidemiology has been about correlations. For example, people who shop at Starbucks 5 times a week to get their $10 coffee, they also have better health outcomes. Is it because Starbucks coffee is so great for you? Or is it because if you can buy a $10 coffee every day, you probably also have better healthcare, better nutrition, better lifestyle, etc. So everything's correlation in human epidemiology, except for genetics. Genetics gives you causation. And that's the excitement, that we now have hundreds of thousands of genetic loci that we might be able to use to understand the mechanism underlying disease. So in this example, I'm showing you this FTO locus, this fat and obesity associated locus, which is the strongest genetic association with obesity, which is telling us that there's something in there that causes obesity. The power of genetics is that it doesn't matter how these variants work. You will know that there's something there. Genetics will work no matter how it acts. The curse of genetics is that after you have discovered those loci, you still have no idea how they work. So what we need is to understand what are the target genes of these loci. How can we develop therapeutics? How can we enable precision medicine and personalized medicine? And that's what we're trying to do in my group. We're basically trying to understand the mechanism through which this FTO locus and thousands of other loci in the genome act. And that's where it gets interesting. If you look at 93% of these genetic loci, they do not perturb the proteins. Here for FTO, I'm showing you 89 common variants that are co-inherited in this haplotype block that spans 50,000 nucleotides between this recombination hotspot and that recombination hotspot. And there are 89 variants, each of which is very significantly associated with the disease, with obesity, but none of which is in fact perturbing the FTO protein. So that basically means that we actually don't know whether the true target gene is FTO or not. And what we showed in our own work in the New England Journal of Medicine a few years ago, in fact, the same year that we had this roadmap epigenomics paper, what we showed is that the true target genes are not FTO at all. FTO is a red herring. FTO has actually nothing to do with obesity, that these variants are in fact hitting IRX3 and IRX5, two master regulators that are sitting 1.2 million nucleotides away and 600,000 nucleotides away. So that I was introduced as the author of the most depressing paper of 2015 in an industry conference. And the reason is that when industry sees 10 genes under this genetic association, they're stuck. They don't know which of these genes is responsible. But when they see only one gene, they're super happy. In this particular case, they were super happy. Collectively, there was more than $1 billion invested in understanding the function of FTO underlying obesity, because the repercussions of obesity are enormous at the societal level. And they all came back empty-handed. And the reason for that is that the true target is not FTO. So that basically means that when you have a causal variant somewhere in here, it's not necessarily that gene. That's extremely important to realize. And that's why my title says circuitry. What we need to do is understand the circuitry for how these loci are in fact targeting nearby genes. So we need to understand what is the causal variant among these 89 that are co-inherited, and sometimes what are the multiple causal variants in the same locus of association. What is the cell type of action? What are the relevant pathways? What is the mechanism? And to understand this, we and many others in our field are basically coupling this power of disease genetics. Here I'm showing you a different example for Alzheimer's with ApoE. The strongest genetic association with Alzheimer's is ApoE4. The most protective variant is ApoE2, and this increases your risk by almost tenfold if you're homozygous for ApoE4. So we start with disease genetics across both common and rare variants, and then we basically say, where do they act? And to figure out where they act, we basically profile the RNA and the epigenome across hundreds of both healthy and disease individuals, samples across the body to basically figure out the tissue-specific effects. Then we integrate the data computationally to predict driver genes, regions, and cell types. And then we validate the predictions using human cell cultures and animal models. And of course, disseminate the results and start the cycle all over again. What do I mean by circuitry? I basically mean that instead of saying there's something going on in this region, we say, well, no, there's 4 SNPs. It must be one of those 4 SNPs. And these overlap very specific regulatory elements. And these elements are active in very specific cell types and tissues. And they contain very specific motifs, which are bound by very specific regulators. And together, these ultimately target very specific genes. So that's what we mean by circuitry, figuring out the upstream regulators, the motifs, the enhancers, the cell types of action, the SNPs, and the target genes. And applying all of this to the FTO locus that I showed you earlier, we basically obtained the circuitry in this paper that I like to joke is about 1 bit of information in the human genome. So this is a C to T alteration. Most of you guys have a T. I carry 2 copies of the C allele. So both mom and dad gave me the obesity risk allele for these locus, which basically causes a cumulative 7 pounds of extra weight as an adult individual. And my cells are basically stuck in the energy storage position, whereas your cells can switch back and forth between energy storage and energy burning. How did we figure out that out? We basically were able to use evolutionary conservation of those motifs to find that there's only one motif, which is evolutionarily conserved, that gets disrupted by this T2C alteration. This is an AT-rich motif bound by this very creatively named AT-rich interacting domain factor, ARIC5B. So ARIC5B sits in a different chromosome altogether. It normally binds this motif. And when its beloved motif is disrupted, then my cells are not able to repress a super enhancer, which is 12,000 nucleotides long, and which is a master regulator of pre-adipocyte differentiation into either energy storing cells or energy burning cells. So many people think of fat as bad. Fat is actually your fat cells are your best friends. They prevent all of these excess calories from being throughout your bloodstream and killing you. They're also extremely helpful throughout evolution, because you can eat on a Monday and still have food left on Thursday, because you can store all of that in energy-rich molecules known as lipids in these white adipocytes, which are white because they're full of these lipid molecules. So that was great throughout evolution, but since 1940, this genetic association has in fact become obesity risk. Before 1940, there was no association. You can look at the Framingham Heart Study. There's no association between FTO and obesity. It's only after World War II when our environment changed. So again, a main message that I want to get across is that genetics is not good or bad. It's well suited for the environment or not well suited for the environment. And for different environments, different alleles are good or bad. If you look at malaria versus sickle cell disease, the same allele is protective against malaria, but risk increasing in sickle cell disease. So it's extremely environment dependent. So we basically found that in this 12 kb super enhancer, repressure could no longer happen. And then we asked what are the target genes through QTL analysis. And we found that IRX3, which is 600,000 nucleotides away, and IRX5, which is 1.2 million nucleotides away, are in fact downstream of this single letter change. And these were previously unknown to have any metabolic roles. And what we showed is that they are in fact deciding whether your cells are depolarizing the mitochondrial membrane through the uncoupling protein 1, UCSP1, PGC1 alpha, and others, to basically relieve the proton gradient. And then instead of forming ATP molecules that then convert into lipids, they basically dissipate energy as heat. So in every thermodynamic equation, you have energy in, energy out, and then energy loss. So what we showed is that by understanding that circuitry, you can understand the mechanism through which this region in the FTO locus contributes to obesity by being able to store or burn calories or being stuck in the store position. And notice the FTO gene doesn't even feature here. The reason why there's a phenotype when you do a deletion study is because you delete the enhancer for IRX3 and IRX5 that sits square in the middle of that gene. So why are we excited about the circuitry? Not because we're academics and we like to see diagrams, but because we're engineers and we like to manipulate diagrams. And by having the diagram, you can basically choose where to intervene therapeutically. And that's the power of genetics. You now have the knobs that you can use to turn the phenotype one way or the other way. So we can increase or decrease the expression of the upstream regulator, increase or decrease the expression of the downstream target genes. And each time it shifts back and forth between lean and obese phenotypes. And we can even use CRISPR-Cas9 genome editing to show that the single nucleotide variant is sufficient to recapitulate the phenotypic difference. And indeed, in primary adipocytes from risk individuals, you have no thermogenesis measured as oxygen consumption rate, both basal and under isoproteinol stimulated conditions. And by altering that single letter out of 3.2 billion letters, so that's as precision medicine as it gets, so one letter out of 3.2 billion allows you to now completely restore the cellular phenotype. And what about the organismal phenotype, where we can manipulate the downstream target genes in a mouse model And basically, under the control of an adipose promoter with a dominant negative form of IRX3, we can basically show that the mice start out leaner. And most importantly, when you put regular mice in a high-fat diet, they gain weight. When you put these IRX3 dominant negative adipose promoter mice, they basically are unable to gain weight in a high-fat diet. So this is very exciting. Basically tells us that we can go from genetics to manipulation. And I showed you one example for FTO and obesity. We published another example a few days ago, or actually a few weeks ago, in collaboration with Li Hui Tai in her lab for ApoE4, the second association that I showed you in the introduction. So this is strongest genetic association with Alzheimer's. We basically looked at single cell profiling of Alzheimer's and non-Alzheimer's individuals, of ApoE4 carriers and non-carriers, of male and female individuals. And what we saw is that in oligodendrocytes, we see this massive change in cholesterol biosynthesis pathways, which are greatly altered. And so it's both transport and synthesis of cholesterol that are altered. And when we looked in cellular models, in organoid models, and in mouse models, we saw that cholesterol, in fact, accumulates in the endoplasmic reticulum of oligodendrocytes. Instead of being exported to the end feet, which are basically coating the neurons with myelin using all of these lipid-rich molecules, you basically have the cholesterol getting stuck. And that basically leads to myelination in ApoE4, in ApoE3 individuals, and lack of myelination in the ApoE4 individuals. So on the same day that our paper appeared, Chris Hemsworth, the actor that plays Thor, the god of thunder in the Avengers, in fact showed that he's not as invincible as you would expect for an immortal. And in fact, he is double homozygous risk for ApoE4. His whole family has a history of Alzheimer's disease. And basically, he's wondering, like all the carriers are wondering, what can we do about it? And the answer is, well, you need to restore cholesterol transport. You need to restore cholesterol to the right place so that you can restore myelination. And indeed, with a cyclodextrin intervention, which is a cholesterol transporter, it's an energy, it's a hydrophobic molecule. You basically show in mice that you can restore myelination. You can relieve the stress from the oxidative stress of all of the lipids being stuck inside the oligodendrocytes. And most importantly, you can also restore cognition. So this is very exciting. So we can basically go locus after locus and start understanding the circuitry, understanding the mechanism, and then start developing, hopefully, new therapeutics. So how do we do this systematically? What we want to do is, number one, understand that circuitry. And that's where reference epigenomes come in. We basically need to understand the circuitry of every tissue and cell type in the human body. You then need to understand how this reference changes from person to person. You need to understand how these are acting at the single cell level and how these are perturbed by genetic variation and how these are acting across multiple phenotypes, across multiple tissues, and then develop methods for manipulating them systematically. So that's what we've done. So basically first, in a wonderful collaboration with Bing Ren and Brad Bernstein and many, many others through the ENCODE community and the Roadmap Epigenomics community, we have basically built reference maps of the human epigenome that allows to look at adult embryonic induced pluripotent stem cells, iPS-derived and primary cells, and across a diversity of modalities between histomodifications, chromatin accessibility, DNA methylation, and gene expression. What does the variation between tissues for a reference set of tissues look like? And to use these, we're basically using these combinations of histomodification marks. So every single state of the chromatin, every single epigenomic state of the chromosome is encoded through some combination of marks, DNA accessibility, DNA methylation. So we can now use these combinations of marks to learn the language of the epigenome. And in collaboration with Jason Ernst, who's now a professor at UCLA, we basically developed ChromeHMM, a tool that allows you to learn de novo across the entire genome the patterns of recurrent combinations of these histomodification marks, and then use them to define different chromatin states, which we then recognize to be enhancers, promoters, transcribed, repressed, etc. So Bing Ren's lab has basically played a pioneering role in understanding several of those. And we were able to sort of put that in the larger context of all of the combinations of chromatin marks. And why is that exciting? Because now we can go across different tissues and look for overlap between the genetic variants associated with different traits and the enhancers active in different tissues. And that's very exciting because we can now see that genetic variants associated with height are enriched in enhancers that act in embryonic stem cells. Genetic variants associated with immune traits act in enhancers that we find in T cells and B cells. Genetic variants associated with blood pressure appear to be only enriched in enhancers active in the left ventricle of the heart. Notice I'm never talking about genes here. This is directly looking at the enhancer overlap in the non-coding regions because that's where the genetic variants are. So by annotating the non-coding genome, we can look at this direct overlap between them. If you look at fasting glucose traits, you basically see that it's very specific to the pancreatic islets, which basically regulate insulin. If you look at cholesterol, you see very specific enrichment for liver, both for HDL and LDL. If you look at inflammatory bowel disease, you basically see both the immune component, as you would expect for inflammatory, and this digestive component, as you would expect for bowel disease. This is very cool, right? Anybody wants to take a bet as to where did we find Alzheimer's disease? Who wants to bet brain? No takers? So very good. You would have lost a lot of money. And I did lose a lot of money. I would have put all my money on brain because that, obviously that's what's affected in Alzheimer's. But it turns out that brain didn't show an enrichment at all. So either our methods was wrong, or maybe our understanding was wrong, or maybe something else. And instead, we got a hint because the strongest enrichment was for CD14 plus monocytes. These are circulating cells, which are from the same developmental lineage as macrophages, the tissue resident immune cells in many tissues, which we call microglia in the brain. So in our brain, only about 5 to 10% of cells are these adapted macrophages that regulate all kinds of brain functions. And no wonder we didn't see an enrichment here because they only make up a tiny fraction of the tissue. So that basically prompted us to say, well, maybe we should be looking at cellular type resolution. And indeed, we sorted cells from the hippocampus and the prefrontal cortex and separated neurons from microglia from oligodendrocytes and showed that, in fact, the enrichment was very specific to microglia. So that basically tells us that we really should be doing all this at single cell resolution. So we wrote a paper basically saying that, well, conserved epigenomic signals in both mice and men show an immune basis for Alzheimer's disease. And of course, the field has really embraced this, sometimes for many decades, but there's a very big role right now in understanding the immune roles underlying Alzheimer's disease. There's a lot of hypotheses about a pathogenic role for Alzheimer's and neurodegeneration more broadly. And indeed, in early Alzheimer's and late Alzheimer's mouse models, we basically found this progressive first activation of immune processes and only later repression of neuronal processes, suggesting that perhaps inflammation and immune dysregulation might be, in fact, the causal component for the vast majority of the genetic associations with Alzheimer's. Not all of them, of course, but most of the common variants, about half of them, slightly more than half of them, appear to be acting in microglia. So we expanded from 127 epigenomes to 134 epigenomes. That led us from 54 enriched traits to hundreds of enriched traits. You can see here a very rich map connecting the two. And that allowed us to now start painting not just a matching between traits and tissues, but also a network where every node is a pie chart, where every trait has multiple associations with multiple tissues. You can see some examples here. If you look at schizophrenia, neuroticism, etc., they localize very specifically in the brain, as you would expect, because they are acting primarily in excitatory and sometimes inhibitory neurons. And that's also the place where you see educational attainment, cognition, etc. By contrast, you see Alzheimer's here sitting with immune cells instead. You can see here coronary artery disease sitting in the middle of heart and a bunch of other internal organs. You see filtration traits associated with kidney. You see liver here with cholesterol and so on and so forth. But I was very interested, we were all very interested in these sort of polyfactorial traits, these traits where the pie chart has many, many different colors. Here's one example. Coronary artery disease, as I mentioned, sits somewhere in the middle of this graph and is in fact enriched for a large number of tissues. If you look at QT interval, you only get heart. If you look at educational attainment, you only get brain. If you look at Alzheimer's, you get mostly immune and some brain. If you look at waist to hip ratio, you start getting more factors involved. Health span, even more. And then coronary artery disease had the most. And we said, well, what if we could use the epigenome to partition the subset of variants that act in liver versus epithelial cells versus coronary artery, and so on and so forth. And indeed, we found that those partitions gave us very distinct biological enrichments. The genes that overlap liver, the genetic loci that overlap liver enhancers are associated with genes playing very different functions from those genetic variants overlapping enhancers acting in the coronary artery versus in adipocytes versus in thyroid cells and so on and so forth. So that's very exciting. Basically allows us to start partitioning polyfactorial traits into each of their factors and start understanding the heterogeneity, maybe even of disease. That maybe we shouldn't be thinking of a single drug against coronary artery disease. Maybe we should be thinking of a combination, which might be even tuned to each individual based on the combination of pathways that are dysregulated in that individual. We can use that information to now go back into individual loci and start asking what are the variants, as I showed you in the first diagram, what are the upstream regulators, what are the downstream target genes, what are the tissues of action. And in this EpiMap paper, we basically predicted these circuits for 30,000 of these variants associated with a huge diversity of traits. So for every one of those, you can basically click on, say, coronary artery disease, get the top loci, and then see what are the top enriched tissues. And for every one of the loci, say, well, where relative to the SNP is that enhancer? And you can see here how many nucleotides away this is. And you can click and see an image that basically tells you what are the SNPs, what are the downstream target genes of the SNPs, how are they linked through enhancer gene correlations, and what tissues are they acting in. And you can find some loci, such as PCSK9, where the mechanism appears to be very liver-centric. EDNRA, very heart and vasculature-centric. And you can find other loci, such as PLPP3, where you basically have different variants acting in both liver and coronary artery disease, coronary artery tissue. And then in both cases, they're linked to the same locus, but with two divergently transcribed genes, one acting in liver and one acting in heart. What are the odds? Well, there's an ascertainment bias. If you take a random locus, the chance that it acts in both is very small. But if you take a disease locus, maybe the reason why it escaped selection in any one of these traits or tissues is because the effect is rather small individually. But maybe the reason why it became genetically associated with coronary artery disease is because when you perturb both together, you have an additive or maybe even a combinatorial effect. So this is one example of both multi-gene and multi-tissue pleiotropy. So this is about building these reference epigenomes to understand gene regulation, but to understand the motifs themselves, I mentioned that we use comparative genomics. So for this, we basically developed a series of techniques that we call evolutionary signatures for understanding the function of any one segment of the DNA based on the patterns of change that that segment has. Conserved nucleotides here are shown in white. We don't care about conservation. What we care about is the specific type of divergence. When there is a mutation, is this mutation amino acid preserving in green or amino acid disruptive in red? And by looking at this very simple signature of codon substitution frequencies, we're able to distinguish protein coding genes with enormous significance and detect coding regions down to just a few nucleotides. Using compensatory changes in the structure of folds, we can distinguish RNA structures. We can find microRNAs using the specific shape of conservation. and we can find regulatory motifs using their genome-wide conservation property and their branch length score over which they're conserved, the number of species over which they're conserved compared to random motifs. So we've built these evolutionary signatures to systematically annotate these reference circuits, and we use them to annotate first protein coding genes. And what did we find? We found that 118 previously thought to be non-coding GWAS hits were in fact protein coding after all. We were just blind to them. Here's one example associated with very fittingly myopia. We basically have a new association that now overlaps a protein coding exon that we just predicted. This was previously thought to be non-coding, but we now have an exon which is alternately spliced and only spliced in retina. And we have an amino acid disrupting change. And this is just one of more than 100 different examples where we can now go back into the non-coding genome and say, well, it turns out it's not non-coding after all. But much more importantly, we can start annotating regulatory motifs systematically across the genome. So we've written a series of papers across yeast, human, flies, and more mammals that basically allowed us to discover de novo the dictionary of regulatory motifs and now start painting these motifs based on their individual occurrences across the genome. Comparative genomics can be helpful for human, which is an enormous and difficult to understand genome, but you would think that maybe it has no power for SARS-CoV-2 that only contains 30-some genes. But the answer is that despite this genome being several orders of magnitude smaller than human, 4 orders of magnitude smaller, we basically have only 30,000 nucleotides, and yet many of the ORFs are still hypothetical for the SARS-CoV-2 genome underlying the COVID-19 pandemic. So at the start of the pandemic, we basically gathered 44 closely related species, analyzed their genomes through our signatures for protein coding genes, and we showed that ORF10, which was previously annotated as an open reading frame in a protein coding gene, is in fact not protein coding at all. It's a non-coding RNA, changing the way that we should be studying its function. We also discovered a new gene sitting within ORF3A that we now call ORF3C, which is encoded in 2 different reading frames, both on the same strand, but offset by few nucleotides. So if the translation starts in this ATG, you get ORF3C. If it starts in that ATG, you get ORF3A. We're also able to use comparative genomics to study the evolutionary context of individual mutations that rose in frequency throughout the pandemic, and to also find genes that evolved much more rapidly during the pandemic than what you would have expected based on their evolutionary speed across recent relatives in the SARS-CoV-virus, SARS-related beta coronavirus family. And for example, the nucleocapsid protein is evolving much more rapidly and specifically associated with a region that we believe to be a neoantigen against the virus that our body can recognize. So this is building these reference maps of the circuitry, understanding the reference epigenome, understanding the reference regulatory control circuitry. How do we now see how this varies across individuals? And to do this, we basically look at the path between genetic variants and disease, which is extremely long. As I mentioned, in height, for example, less than 1 millimeter of your height. These are very small effect variants and extremely polygenic. But by looking at these intermediate effects, we can actually break up that long path and start understanding how these variants act in specific tissues, in specific regulatory elements, in specific genes, and for specific endophenotypes. The goal here is that by breaking up this very long path and this very polygenic architecture into smaller subsets, we can basically now start breaking up this polygenicity and start seeing much stronger effects. So basically, maybe that SNP has a much stronger effect at the level of gene expression or at the level of enhancer activity. The downside of these intermediate steps is that there's now bidirectionality. Some of these changes might be, in fact, downstream of disease. So to do this, we basically have to look at mediation. We need to understand how genetic variation is mediated through these epigenomic effects or these transcriptomic effects in order to understand causality. So if you look at genetic correlation with anything, you get unidirectional arrows. If you look at anything else, you get bidirectional arrows. Some of those changes could be downstream of the disease. In fact, some of them could be combating the disease. Just because you see firefighters every time there's a fire doesn't mean that they're causal. And if you suppress the firefighter gene, you basically will get more fire. And some of these genes that go up with Alzheimer's might in fact be pushing against Alzheimer's. And if you repress them, you might get more Alzheimer's. So to understand causality, we need to first look at this arrow and look at how we can predict epigenomics from genetics. So here we're looking at the genotype of an individual at conception, and then the epigenome of that same individual at 93 years of age. And what you can see is that if I know your genotype at birth, I can predict your epigenome at that locus with very good certainty. And we can do this across hundreds of loci. We have 50,000 significant methylation QTLs. And that allows us to now start breaking up that long path of GWAS, genome-wide association study, to perhaps MWAS, methylome-wide association studies. But notice now we have a bidirectional arrow. Instead, we can look at methylation QTLs and start predicting the methylation, and then use the genetically inferred methylation, this imputed methylation, and then correlate that with disease. This is IMWAS. And what we have now is a unidirectional arrow, because this methylation is only predicted from local genetic variants. And that basically says that it's much more likely to be a causal factor for disease. So using this, we can go from no significant loci in all of chromosome 6 into half a dozen such loci, which are now significant because, number one, we're testing fewer hypotheses. We only look at methylation variables, not at every genetic variant. And number two, we're combining multiple SNPs together to predict methylation, which is in fact more robust. Applying this at the transcriptome level and at the methylation level, we can look at the path of causality across multiple variables and start inferring driver genes, the directionality of effect, the tissue in which they're acting, because that's where we measure the expression, and also new loci, whether there was a significant hit or not at that locus. And the reason for that is that we can use all of these new variables, fewer hypothesis tests, and we can now find both risk-inducing and protective effects associated with Alzheimer's disease. But all of this is happening now at the tissue level. We need to go to single cell level, as I mentioned earlier with the microglia example. So we've basically started profiling at single cell resolution a large number of cohorts across Alzheimer's, frontotemporal dementia, Lewy body dementia, ALS, Huntington's disease, psychosis in AD, schizophrenia, bipolar disorder, Down syndrome, autism, depression, suicide, PTSD, and aging more generally, with dozens of individuals in each of those, sometimes up to hundreds of individuals for the same trait, and then looking at multiple cell types, multiple brain regions, and looking at both RNA and DNA accessibility using single cell RNA and single cell attack. In the first paper that we published a few years ago, we basically looked at only 48 individuals for early and late pre-symptomatic, early and late Alzheimer's. You can see here the pathogenic signatures. You can see here the cognitive signatures of Alzheimer's. You can see neurodegeneration through amyloid plaques and neuritic plaques, amyloid beta accumulation, neurofibrillary tangles, and cognition decline. You can see here high cognition, then cognitive loss, and eventually loss of cognition in those individuals. So what did we find? We found that, in fact, we could cluster these cells into subclusters that very often associated with AD versus non-AD, but also with male versus female individuals. And the female individuals had many more of these risk-associated categories, suggesting that perhaps the transcriptional alterations were stronger in female individuals. We also saw that there were early changes that were extremely cell type specific, but late changes were much more shared between cell types and very often associated with damage response rather than these early changes, suggesting that perhaps early Alzheimer's might be the place to look for these cell type specific causal mechanisms rather than just the response to disease. And generally, we found that thousands of genes were differentially expressed between men and women prior to AD, but in fact, that number doubled for excitatory neurons in AD, suggesting that men and women are different to start with in their expression patterns in the brain, but that in the context of AD, they're even more different. And again, suggesting that perhaps we should be thinking of sex-specific therapeutics for many of these dysregulations that might be happening. In particular, we found that white matter loss was more pronounced in female individuals after seeing that myelination pathways were activated in male individuals, suggesting perhaps a protective effect. And then we went and indeed confirmed in the pathology that this was the case, and that was not previously observed. We have seen scaled from 84,000 cells in 48 individuals to 1.6 million cells across multiple brain regions of 48 individuals. and more recently across 430 individuals in the prefrontal cortex. So basically, we've looked at prefrontal cortex, midtemporal cortex, angular gyrus, entorhinal cortex, thalamus, hippocampus, and mammillary body, enabling us to discover a huge diversity of excitatory neurons, inhibitory neurons, and also subclasses of astrocytes, oligodendrocytes, and even microglia. Here you can see how much the inhibitory neurons vary between cell types, and also that even glial cells, in this particular case, astrocytes, vary dramatically. We found a total of 30 different subtypes of excitatory neurons, enabling us to actually do very, very fine grain resolution. And across our 1.6 million cells, we can now start looking at temporal models of AD progression and looking at cells that are transcriptionally more similar to controls, cells that are transcriptionally more similar to Alzheimer's, and a whole progression between the two, enabling us to see what are the genes that are the most dysregulated early, middle, or late in Alzheimer's disease. From the single cell expression patterns, we're also able to infer the anatomical positioning of those neurons. We basically looked at these pyramidal cells versus granule cells in the hippocampal formation across the subiculum, the dentate gyrus, CA1, 2, 3, and then different layers of the entorhinal cortex. And what we saw is that these cells clustered very precisely according to their anatomy. We have used this to start now building deep learning models that allow us to predict from the expression patterns of individual cells, their 3D positioning, using the mouse as a training set, and then applying that to human, and to also deconvolve these Visium experiments of mini bulk multi-cell expression patterns into individual cells. And when we do that, we see that for Purkinje cells, for example, in SCA1, in this ataxia, we basically see that the Purkinje cell markers are not differentially expressed, but then the Purkinje cell signatures after deconvolution are greatly altered in SCA1. You can see here in SCA1, you see this great loss in organization of the cerebellum, when in fact, you see this very nice organization in the control individuals. We've also used Resolve, a new technology that is looking at subcellular resolution to start positioning individual genes in different spots. And you can see here extremely precisely in coronary artery disease, this positioning in the intima, in the lumen, in the media, associated with specific marker genes that we're discovering from our single cell data, and then using as markers in the 1,000 probes that we get in Resolve to basically get at, number one, the markers of different landmarks in these images, but also the differential expression in the context of CA1, of CAD. And we're basically looking at these regulatory networks that are emerging from these spatially resolved differentially expressed genes, and also these spatially localized inflammatory changes in these immune cells that we call foam cells that are thought to underlie coronary artery disease. And our goal is to ultimately bridge the gap between imaging and expression, the same way that we're now building these common projection models for single cell attack and single cell RNA in this sort of deep learning joint embedding space that allows us to use these attention architectures to start localizing the regions of the genome that the algorithm pays attention to as candidate enhancers linked to those target genes, linked to the upstream regulators, and enabling us to learn translation tables between the two. And our goal ultimately is to be able to also do that for imaging. Having so many cells allows us to also learn modules of coordinated changes in the gene expression patterns of those cells, enabling us to find genes that are coordinately active. And those genes, in fact, reveal a large number of modules of very biologically coherent subsets of genes. We can use these modules to now take these rare cell types like microglia and understand how each of these modules is altered in association with different phenotypic signatures of Alzheimer's disease. And we can also find coordinated changes between brain regions that are known to be connected. For example, here between the dentate gyrus and the layer 1 of the entorhinal cortex, we basically know that there's an anatomical connection there between these different regions. And indeed, we find that these regions show coordinated changes between individuals. suggesting that in fact we might be able to even infer the anatomical connectivity between these different regions through neuronal projections from the single cell expression patterns. In a paper that we posted in medRxiv a couple of years ago, we basically looked at schizophrenia changes that are associated with genetic loci that are known to play causal factors in schizophrenia. And for each of these loci, using high-C data, we were able to map proximal genes that are showing altered expression, either increased or decreased, giving us, again, directionality of effect, and also the cell types where they appear to be dysregulated. Using the combination of those markers, we can basically start predicting which individuals have schizophrenia-like transcriptomes and control-like transcriptomes. And you can see this gradient between the two, where all of the individuals here on the red appear to be schizophrenia cases with a number of exceptions. This exception right here is phenomenally interesting. That person was never diagnosed, but his son was in fact diagnosed with schizophrenia, suggesting perhaps either protective mechanisms in the father, or perhaps different diagnosis or environmental factors. And conversely, individuals that show very control-like patterns, but are in fact diagnosed with schizophrenia, we can now start thinking, well, are there different subtypes of schizophrenia? Are there perhaps different paths transcriptionally? And what we're finding is that these individuals are enriched for this SZTR schizophrenia transcriptional reversal cell state that we identified, posing an intriguing hypothesis that maybe it's not just gene expression pattern that we should be looking at, but also cellular state abundances. Using this, we can start predicting upstream regulators for these dysregulated genes, and then these group into three subsets. Basically, there's synaptic signaling, synaptic organization, and also neurodevelopment. And we can find upstream regulators associated with those and how they're grouping into modules. And one module in particular associated with MEF2C, a master controller of cognition, that also came up in our analysis of educational reserve and cognitive reserve from early educational enrichment in both mice and humans. And this is very specifically overlapping a module that was previously discovered in PsycEncode, suggesting that several of these genes that are associated with schizophrenia, with depression, and also with both, might in fact be playing that role. And these appear to be acting not just early and late, but not just early or late, but also both. So this is psychosis without AD. We also have psychosis with AD. So we basically looked at the subset of individuals that develop psychosis versus the subset that do not, in collaboration with Robert Sweet and Julia Koffler at UPitt. And what we found is this massive repression in excitatory neurons of psychosis individuals, suggesting that psychosis is not an overexcitatory state, but it is in fact a repressive state where less neuronal activity is happening, even in excitatory neurons. And there's several pathways that are enriched here, including ribosome biogenesis, neuronal projection, autophagy, secretion, signaling, and energy. So again, in schizophrenia, sometimes it has been argued that there's overactivation, but here we actually find repression associated with psychosis. In a paper that we just submitted last week, we basically looked at the differences in ALS individuals and FTD individuals. Why did we study them together? Because they're both associated with C9orf72, and also because they sometimes run in the same family. And sometimes individuals who are carriers of the same exact mutation will manifest as cases or controls for one or the other disorder. Hello, Trey. If it wasn't you, I wouldn't have interrupted, but it's a pleasure to see you. No, but I'm so glad you're here. So what was really remarkable is that we were using the motor cortex in frontotemporal dementia as a control region, and the frontal cortex in ALS as a control region, expecting that we wouldn't see anything there. But in fact, we were struck that both showed changes in both. And in fact, the two regions appear to be quite orthologous in their cell types. With one exception, these long-range projecting excitatory neurons, known as BET cells in the motor cortex or von Economo cells in the frontal cortex, are in fact splitting into 1 to 2. So basically there appears to be two different subtypes here in the motor cortex of BET cells. And only one of them appears to be very severely affected in ALS. But what's really remarkable is that the pathways appear to be the same, the cell types appear to be the same, and in fact, both traits showed dysregulation in both brain regions, suggesting that there's really much more than meets the eye in many of these neurodegenerative disorders, and there's maybe many more overlapping regions of alterations. And in fact, they're very often categorized in a continuum between, Alzheimer's, even prion disease and PSP and CBP and so on and so forth. So we are now actively pursuing separate cohorts in each of these disorders, and ultimately hope to recruit some awesome postdocs to look at the similarities and difference between them. In a paper for Huntington's that we just published a few weeks ago, actually 2 weeks ago, we saw this difference in identity between indirect pathway and direct pathway spinal projection neurons, which are sort of very distinct prior to HD, but lose their distinction in Huntington's. In a grant that was just funded, collaborating with Rudy Tanzi at MGH, we're basically looking at AD in association with different pathogens. So I mentioned earlier that this immune dysregulation underlying AD is very often thought to be possibly an environmentally triggered condition, and maybe there's a connection there with microglia. But there's another even more foundational hypothesis that amyloid itself might be playing immune roles in the brain by trapping pathogens. And indeed, experiments in mice that were carried out by Will Eimer in collaboration with Rudy Tanzi showed very elegantly that mice exposed to pathogens will develop more amyloid, and mice with more amyloid will be more protective against these pathogens. To make a long story short, we basically looked at CMV plus and minus individuals, HSV1 plus and minus individuals, and then we found dramatic alterations in multiple pathways associated with immune functions in microglia, in the vasculature, and in several other cell types. COVID itself has been thought to have an association with long brain dysregulation and disruption. So basically, you've all heard about long COVID. Some individuals basically show neurocognitive symptoms that last a long time. So we basically looked at the impact at single cell resolution of individuals with and without COVID. And what we found is, number one, this increase in astrocytes. Number two, this decrease in expression of excitatory and inhibitory neurons. And this enrichment for, as you would expect, RNA metabolism and RNA processing genes, which you expect to be the result of the virus, but also this decrease in synaptic organization, neuron projection, cell junction, and so on and so forth, suggesting indeed a decrease of neuronal activity. And very interestingly, even though the changes are happening in all of the cell types, the enrichments appear to be strongest in microglia and in vascular cells, as you would expect as the point of entry of the virus. I mentioned at the beginning this ApoE4 paper that we just published in Nature a few weeks ago, looking at these cholesterol changes associated with a genotypic alteration. So now we can look at individuals that have ApoE4 but do not show Alzheimer's disease. And indeed, even the non-AD carriers show cholesterol transport dysregulation. Again, enabling us to now look at the cellular effects at the genetic level rather than all the way to the phenotypic level. And in the same spirit of ApoE4, we can look for rare variants in multiple other genes associated with Alzheimer's disease. And we can go and gather those individuals containing those rare mutations and then see transcriptionally how are those mutations coalescing across the circuitry of the cell. In a paper that we are sending the revision in a few days in collaboration with Na-Sun, we basically looked at multiple subtypes of microglia associated with our about 200,000 microglial cells that we could sort from these 1.6 million overall cells. And we were able to define 12 different subtypes of microglia, including homeostatic microglia, age-related, inflammatory, which are showing some of the most strong dysregulation in Alzheimer's disease. In a paper we published in Nature exactly 1 year ago, we looked at the vasculature of the brain. This is 0.3% of the cells, and we're able to map the zonation gradient of venule to arterial and capillaries of the vasculature with distinct signatures. And we now have a paper that was just accepted and is basically looking at the difference across different brain regions for these vascular cells, and also in Alzheimer's associated dysregulation, and the regulators and the cell-cell communication between them. At the epigenomic level, we basically have the ability to now distinguish individual cells through single omic data, through single cell attack or single cell RNA. And we are now looking into multi-omic data where we have both from the same cell, and the data is remarkably superior, enabling us to now start annotating many different subtypes of these epigenomic signatures for subsets of excitatory and inhibitory neurons. This is from attack-only data, but the joint data looks much, much nicer still. And that allows us to now start mapping genetic variants into specific cell types and upstream regulators associated with multiple variants in these subtypes. And we're also looking at the mutations that are accumulated from whole transcriptome sequencing using SmartSeq and more recently 10x and Nanopore to basically start calling mutations directly from the single cell RNA data. And what we're finding is that the AD individuals are showing many more mutations called than the non-AD individuals across multiple cell types. And in particular, we're finding that this subset of senescent cells are in fact showing an accumulation of mutations possibly associated with DNA damage. Those mutations do not occur randomly. They're enriched in biological pathways associated with Alzheimer's disease, enabling us to now start looking at perhaps this genotype-phenotype relationship, not at the individual level, but at the cell level. Because for those cells, we know that they contain increased mutations, and we know that they are transcriptionally clustered. So here you can see that the cells with more mutations are also showing common transcriptional profiles, indicating this genotype to transcriptional phenotype relationship. Lastly, we're seeing this global epigenome erosion in these late-stage individuals for AD, where the open regions become closed and the closed regions become open, specifically in late AD, not in early AD. And what we're seeing is this global shift in the distribution of these erosion scores, where we see a modest degree in the non and early individuals, but no difference between them. And then this dramatic shift in the distribution. And this is basically possibly a mark of the genome detaching from the nuclear envelope, which helps hold together her heterochromatin and distinguish these active from repressed domains. And you can see here individual cells sorted based on their erosion score, and this change from accessibility to inaccessibility and from inaccessibility to accessibility. This is looking at one phenotype at a time, but now that we have all these data, we can start looking at multi-phenotype integration. And in particular, with electronic health records, we're able to look at lab tests, ICD-9 billing codes, prescriptions, DRG codes, and doctor notes, and build them together in a multiple hierarchical model, a multimodal hierarchical model. You can scan any of these codes to get the corresponding papers. And most interestingly, we're now looking at Alzheimer's disease and how we can understand that heterogeneity of patients that we can see through the clinical record, through the phenotypic variables of those individuals and also the transcriptional variables of those individuals. The idea here is to go back to those transcriptional hallmarks that I mentioned earlier. Can we understand heterogeneity of disease by focusing in at the molecular level at how each pathway is dysregulated in a different subset of individuals? And can we find that similar pathways are repeatedly, recurrently dysregulated in different subsets of individuals, and that those are in fact enriched in biological processes, and those are in fact enriched in distinct phenotypic manifestations of the disease, suggesting that some transcriptional hallmarks are in fact associated with early stage versus late stage of Alzheimer's, some with more tau pathology versus amyloid pathology. And we can now start grouping individuals into different subtypes of Alzheimer's disease based on their transcriptional signatures, which are then also recapitulated in their phenotypic signatures. And we're now using genetic predictors to infer which of these subtypes an individual is likely to fall in order to start prioritizing therapeutic suggestions for the corresponding individuals. We're also looking at these biomarkers associated with disease and how, in fact, sometimes there appear to be inversely associated in very sharp thresholds. And some of the reason is that the biomarker itself is not only predictive of the disease, but also predictive of the treatment that will then reverse the disease. So that basically suggests that we need these causality models to understand these potential reverse causality in some of those cases. And we're also looking at longitudinal progression of disease, including cancer in this particular example, where you can actually trace the same individual across multiple clones, across multiple biopsies, and the specific mutations that are being selected in each of those. And we now have a cohort of more than 100 individuals with single cell profiling before and after immunotherapy to basically look at, number one, predictors of immunotherapy response in metastatic melanoma, and number two, what actually changes for those that respond to immunotherapy versus those that do not. And in particular, we're finding these epigenomic alterations where these active enhancer regions are getting shut off in a large number of individuals, predicting an upstream regulator that we can now start using to use as a combination therapy along with immunotherapy. And indeed, you see that there's almost no recurrence when you use this combination treatment in a mouse model. A few weeks ago, we had another paper appear in Cell Metabolism looking at the multi-tissue effects at single cell resolution of exercise and diet interventions, enabling us to now start understanding the pathways that are altered and tying back in with this FTO story that I mentioned earlier, where we can basically see that exercise rewrites your metabolic and it rewires your metabolic tissues to actually burn more calories. You see changes happening in the mesenchymal stem cells that then give rise to basically both fat burning and fat storing cells. There's reprogramming of those stem cells, there's changes in cells of communication, and there's a lot of immune processes that are sort of mediating these changes between exercise and obesity across multiple tissues. I mentioned a few examples where we are perturbing these genes systematically, but we've also built new technologies for being able to manipulate multiple genes simultaneously and being able to construct these modular and programmable CRISPR-Cas9 constructs that allows us to either use Cas9 for editing or knocking out genes or promoters, and dead Cas9 or deactivated Cas9 with P300 or CRAB to basically bring in activators or repressors. We can induce those in iPSCs and then differentiate them into different cell types to look at the cell type specific manifestation of those interventions. We also develop tools for profiling millions of tests in a single experiment across many different cells. How? By chopping up DNA directly from accessible regions, inserting it into these self-transcribing constructs, which are acting as their own reporters, and then using that to map activity in every single segment, enabling us to now start tracing activity to individual nucleotides and individual regulatory motifs, which are matching conserved regions and are enabling us to start interpreting genetic variants that are underlying disease. We're testing several of these variants in high throughput way using massively parallel reporter assays in collaboration with Andreas Fenning, former member of my lab, who's now a professor at CMU, who's now inducing these mutations associated with these alterations in the brains of mice to basically see how they're acting not in an episomal construct, but how they're acting in the context of a whole human brain. And then lastly, we have collaboration with Marinka Zitnik and Brad Pettelute to start designing these chemical structures directly from the deep learning graphical neural networks to use this geometric deep learning approach to start designing mimics of the domains that we're finding to be underlying this. In this particular case, this is IRX3, and how we can use biological mimics of these domains to start developing new therapeutics and start targeting them in a cell type specific way. So that's what I'm going to leave you with. Basically, we look at, number one, reference epigenomes to understand the basic circuitry. We combine genetic, epigenomic, and transcriptional variation across individuals and in the context of disease, both at bulk and single cell resolution, to understand these genetic effects across multiple phenotypes, across multiple tissues, and to disrupt them systematically. We are hoping that this will sort of really lead to a transformation for personalized medicine, understanding these hallmarks, being able to use individualized treatments. And we really need a very broad coalition to do this across, biology, computer science, chemistry, and both within and outside academia. So again, I'm very privileged to lead this amazing team of individuals. This is at our lab retreat. About half the people are from the lab, the other half are collaborators who joined us for the whole day of brainstorming. These are the people who are doing the work. I try to acknowledge them with pictures in each of the slides. And again, if you go back through the recording, you can see there's QR codes on every slide that you can use to learn more. So I'll stop there and any questions.
That was spectacular. I think many questions. Everyone want to You can shout out.
Go ahead. I can see you looking back. No.
Do we have people online to have questions?
Yeah, I can ask a simple question.
Yeah, go for it.
Yeah, so as you start to figure out particular categories, that might be tissues or cell types,
Yeah.
et cetera, that are important for a disease, and I think you have the example of coronary artery disease,
Yeah.
something like that, vascular disease. Would it be to take out PRS, which ordinarily is kind of like equal
Beautiful,
to lots
beautiful.
of little variants,
Yeah.
right? And instead to say this person has a strong PRS, say for the liver specific factors that coronary artery disease. And maybe those would have super additive or other kinds of insights that you would get.
Yeah, absolutely.
It's giving them all equal weight.
No, this is exactly what we're doing. So basically, here I mentioned that many of our polygenic scores can now be computed at the pathway level. And let's see, there you go. So we are using genetics to start predicting the total global dysregulation. So I'm with you completely. So we're doing this in multiple ways. We're doing this at the pathway level, using a gene-centric approach as to what pathway the gene belongs in. We're also doing this in a cell type specific way by looking at all of the enhancers that are active in every tissue of the brain, in every cell type of the brain, and then calculating the PRS specifically for those. Yeah.
And what are the consequences of doing that?
It's working remarkably well.
Yeah.
So basically we're able to classify individuals quite accurately, and many of these individuals, when we sort of ask, do they show distinct phenotype based on their transcriptome? Based on their genetically predicted transcriptome dysregulation, then indeed they show sort of anatomical changes that are very consistent within the groups. Yeah.
I have a really relates to the polygenic score.
Oh.
The pathway polygenic score that you just talked about, and also when you take genetic risk and define the cell types that are enriched in various different disorders.
Yeah.
I somehow miss how you're connecting the SNPs to cell types. In this case,
Yeah.
Are you taking the enhancer itself?
Correct.
Intersecting SNPs with enhancers, or are you taking regional enhancer densities and looking for enrichment?
Yeah. So I have an explanatory slide that I didn't include, unfortunately, but basically here we're looking. Here I mentioned that we're looking directly at the genetic variants and the enhancer active in that cell type. So this is a direct overlap.
This is a direct intersection between
Correct.
associated SNP.
Correct.
And enhancer.
Correct. And of course, the statistics underlying all that are very tricky because there's multiple ways to interpret this philosophically. You could say every SNP matters equally because they're there. They could disrupt these enhancers. So if I have in the same haplotype block 3 SNPs in 3 different enhancers, then that increases my power. Or there's a different philosophy that could say, well, maybe there's only one causal SNP, and then I have to sort of prune out all of the other SNPs and correct for that. So, there's different ways of doing this, and there's something called the multiple enhancer variant hypothesis or MEV hypothesis that I think Corradin is the first author, and then Peter Scorchese is the last author. That basically has that argues that in the same way that I mentioned here with the sort of pleiotropy, where you have multiple variants possibly acting in multiple tissues, where they're basically saying, well, the chance of having a pathogenic variant and then gaining another pathogenic variant in the same locus could be very rare, because why would selection work that way? But in fact, it's the other way around. Every locus gets multiple hits completely at random. But when those hits move in different directions, you don't get a GWAS hit. When they move in the same direction, according to Poisson probability, you basically have, you're more likely to call a GWAS hit there. Oh, I'm sorry. Yeah.
So you think that your tissue enrichments in Alzheimer's SNPs are driven by the fact that there probably are many causal SNPs across the region, and you're getting multiple intersecting SNPs across multiple enhancers?
In part, yes, but the enrichments are very, very strong, whether you use the top SNP, whether you use the most overlapping SNP, whether you use LD score regression to basically regress out the shadow effects of all of these SNPs in LD, where a SNP with many LD partners will basically be boosted, and therefore you can correct for that. You can use that. You can see them when you use all of them, one of them, the max overlap, or the max score.
And last related question, is that from all genome-wide significant SNPs, or do you take some, like a polygenic score, or do you kind of just weight, you just weight the SNPs and then look for enrichment?
So we've done in a bunch of different ways. You can just take the genome-wide significant. You can go to the sub-threshold SNPs, and we had a paper in Eli back in 2018, I think. Xinchen Wang was the first author, which was basically actually going and testing some of these 10 to the minus 5 SNPs and showing that indeed they are biologically meaningful when perturbed. When you use epigenomic marks to basically boost sub-threshold loci, we show that indeed some of them are functional. And again, the field is realizing that many of them will be functional. In a statistical way, you can basically see enrichment all the way down to nominal p-values, which are, 5 orders of magnitude from significance. You can still see biological enrichment. And then there's this omnigenic model that Jonathan Pritchard and his group are arguing for. But basically, in many of these analyses, for studying individual loci, we look at genome-wide significance. For studying enrichments, we very often go to 10 to the minus 5, and then we include many more sub-thresholds, some of which will be fake, but there's sufficiently many real ones in there to further increase our power. I hope that answers. And also there's like threshold-independent ways, very similar to GSEA, gene set enrichment analysis, that basically allow you to look as you go down the range of sort of scores, how does that enrichment shift? All great questions. Shane, can you pass it behind you? Jonathan? Microglia. Yeah. Yeah. Yeah.
This is a disease that doesn't essentially actually our majority of the population at the age. I wonder how the vision of sleep sensor vision is that disease could be actually what can you solve through all that way?
Yeah. No, it's a fascinating question. So for schizophrenia, you have an obvious selection disadvantage, because those individuals in their teenage years and sometimes in early adulthood, before they have kids, they will be, so detached from society that they're, very rare to have children and to sort of certainly bring up their children in sort of successful ways. If you look at infant mortality, up until, 1850, 1900, infant mortality was enormous. People would have a lot of kids so that some of them survive. It is possible that the longevity and the health span of the grandparents could actually have an effect on the longevity, on the survival probability of the grandkids. So if grandma is demented, you might not have as much chance of survival for the great offspring. So it is not extraordinary to think of selection acting that way. That's one possibility. Another possibility is that the same pathways that will lead to neurodegeneration are also leading to other phenotypes in other tissues that might not be necessarily associated with Alzheimer's. So, for example, Alzheimer's has sometimes been called type 3 diabetes. And we see this very huge connection with lipids. I mentioned sort of lipid and cholesterol transport. underlying myelination and cognition. And maybe some of these dysregulation in lipids could have other reproductive, disadvantages in other places. I think in general, it's just a great mystery as to why the heck do humans live so long? Why the heck are humans able to process so much information in the beginning of the 21st century when in fact our brains haven't changed that much since 150 years ago? So I don't know. There's a very big conundrum as to sort of what is this, what is evolution really selecting for? Is there sort of a more general selection for robustness that is over-engineered, if you want to use a sort of, anthropocentric term, relative to sort of the needs of the vehicle, into its late years. But the last aspect of your comment was about the fact that they're immune cells. What does that have to do with anything? And in coronary artery disease, in obesity, in exercise, in Alzheimer's, in schizophrenia, in psychiatric disorders, in depression, we're finding immune cells everywhere. I mean, of course, cancer and immune traits and autoimmune traits, I'm not going to mention, but there's all these traditionally non-immune traits that have immune pathways associated with them. What the heck is an obvious question. And we wrote a review for Neuron recently. It was called a preview because it was associated with another article. But basically, in that review with Nassan and Manolis Kellis as the two authors, we basically argued that of course, there's this observation, but we also discussed how, number one, there are immune genes that are acting both in immune cells and also immune genes that are acting in non-immune cells. And more generally, immune cells, such as macrophages, are tissue resident. M1 versus M2 macrophages in obesity can determine whether, you have inflammation associated with obesity and this lipid accumulation, or whether you are sort of lean and no inflammation. And this ratio of the two is, in fact, a huge predictor of obesity. And the situation here is the following. We basically have immune processes and immune cells and immune processes outside immune cells being co-opted for many different biological processes inside the body. Why? Because these immune genes are incredibly versatile. Two possibilities. One, their initial function is immune. The second possibility is that their initial function is in fact not immune, but it's just we gave them an immune name because we discovered them in immune cells first because immune cells are easier to study. That's another possibility. But regardless of, the chicken and egg direction here, the fact that immune genes and cells are underlying so many different non-immune disorders, in my view, could be associated with the fact that every time we have a pandemic, prior to modern vaccination, etc., every time we have a pandemic, a huge chunk of the population dies off. If you look at the Black Death in Europe, you basically have 30+% of the population that just died. That leaves a giant hole in the optimization of the genome where these immune processes were perhaps optimized for some functions that are non-immune. And now they're just wiped out because they weren't compatible with defense against a particular pathogen. And you basically have this inability to fully optimize your genome for the things that are the most environmentally associated, where this environment is not something that we as a society shape progressively, but these are just viruses or bacteria coming in and killing off a huge chunk of the population. So anyway, there's like massive selective forces there where the environment could be pulling all of these variants away from optimum. And therefore, these functions that have been co-opted are now suddenly manifesting as diverse disorders. Oh, great. It should only take me 10 minutes. We only have the room until 1:30, so we have to. Thank you. Thank you, guys. Thank you. And if you have awesome graduate students finishing, send them here for postdocs. Lots of cool projects. Thank you, guys. Bye. Bye, everyone. There's 45 people still online. Thank you, everyone online. Bye-bye.